Good morning. I am your host, Facundo Batista, and together with Richard Young and Callum Overhaul, we have organized this MIT course on COVID-19, SARS-CoV-2, and the pandemic. On behalf of the three of us, thank you for joining us today. As you know, the purpose of this course is to learn what we know about the virus and the pandemic from the best scientists around the world. And today, I have the great honor to introduce to you Professor Arup Chakraborty. Dr. Chakraborty work centers on defining mechanistic aspects of the adaptive immunoresponse to pathogen and how to harness this understanding to help design better vaccines and therapies. Arup's predictive computational theoretical work has impacted both experimental basic immunology and infectious disease research, especially contributing to our understanding of T-cell signaling and pathogen specificity, the immunological vulnerabilities of HIV, and how this influence rational vaccine design. A hallmark feature of Arup research is the close synergy and collaboration between his lab, which focus on theoretical and computational studies, and investigators led by experimental and clinical immunologists. Dr. Chakraborty was born in India and immigrated to the United States after an undergraduate degree at the Institute of Technology in Kanpur. And after earning his PhD in chemical engineering from the University of Delaware, he did his postdoc studies at the University of Minnesota. Dr. Chakraborty became a faculty member of the University of California, Berkeley. But in 2005, he moved to the MIT, where he is the Robert T. Haslam Professor of Chemical Engineering, Physics, and Chemistry. Dr. Chakraborty is only one of 15 institute professors of MIT, and he was appointed the founding director of the MIT Institute for Medical Engineering and Science, and he's one of the founding members of the Reagan Institutes that belong to MIT, Harvard, and MGH. Along his prolific career, Dr. Chakraborty has received numerous prestigious awards, and indeed, to remark, are 6 teaching awards for his classroom teaching, and is one only of 25 individuals who are members of 3 branches of the US academies: National Academy of Science, National Academy of Medicine, and National Academy of Engineering. So Arup, we are delighted that you are with us today, and we are looking very much to your talk. Thank you very much.
Thank you, Facundo, for the very kind introduction. I hope everyone can hear me. So I have been asked to speak about the history of pandemics and vaccines. I am not a professional historian, so I apologize in advance for the lack of rigor in what is to follow. The human story is inextricably linked with the suffering caused by infectious diseases. And indeed, the written record since antiquity is replete with references to plagues and contagion. As just a few examples, the plague of 541 AD killed the equivalent of half the European population. In the 14th century, the Great Bubonic Plague, which likely started in China and then spread to Eastern Europe and thence through Italy to the rest of Europe and then on to the Middle East. The plague of the 19th century killed almost 10 million people in India, and there were famous outbreaks in Hong Kong, San Francisco, and other places. Cholera is a waterborne disease that causes severe diarrhea and dehydration, often leading to death. In this period of 50 years, there were many worldwide pandemics that all began in the Indian state of Bengal, where I was born. To continue this story of suffering, the 1918 influenza pandemic, about which we have all heard recently quite a bit about, killed about 20 to 50 million people, many more than the casualties of World War I, which was about 15 million, including 6 million civilians. And indeed, it is believed that the end of the World War I was hastened by this influenza pandemic and the havoc that it was causing. In the 20th century, there were flu pandemics, and most recently in 2009, the HIV pandemic continues And there have been roughly 75 million infections and most likely about 40 million deaths due to complications from AIDS. And while it is largely under control because of antiretroviral therapy in many parts of the world, in some parts of the world, like in South Africa, there are 1,000 new infections every day. And of course, COVID-19 is the most recent example of a pandemic that has caused so much trouble. Now, influenza, HIV, and SARS-CoV-2, the virus that causes COVID-19, are all examples of viruses, which have especially caused havoc, particularly in the post-agrarian world. When our ancestors learned how to grow crops, they started living in larger communities. So viruses that could be spread easily by casual contact became easy to transmit to many others. Furthermore, the virus could lead to a disease that was reasonably deadly because it did not get extinguished, because in a large population, new births, and interactions with people in neighboring communities with whom another community traded led to a constant supply of susceptible people. We also started to domesticate animals, and so viruses that circulated in the animal population began to infect humans. Thus, it came to be that diverse viruses that spread by casual contact and which can be reasonably deadly began to circulate in humans. All of this was exacerbated by population growth, the Industrial Revolution, and urbanization. It is instructive, indeed striking, to look at how the world's population, but also the population density, changed in the 1,000 years since 1000 AD. Now, the reason we survived as a species is because we have an adaptive immune system, which is remarkable in that it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes. Imagine how amazing that is, that a person who gets infected with SARS-CoV-2 today can mount a bespoke, tailored response to this particular virus, which evolved after this individual was born. To roughly see how this works, consider infection with a virus. When a virus infects a host cell, it hijacks its transcriptional machinery, enabling the synthesis of new viral proteins and the assembly of many new virus particles that can go on to infect other cells. Now, B cells are important players in adaptive immunity. We have about 100 billion B cells, and most B cells have a receptor on their surface that is distinct from that on the surface of another B cell. If the receptor on this particular B cell can bind sufficiently strongly to the spike proteins of this particular virus, then it gets activated, it starts to multiply, and then a Darwinian evolutionary process occurs in a very short time, which ultimately leads to the synthesis of even higher affinity receptors, which are then secreted in soluble form, and that is what we call antibodies. These antibodies can then bind specifically and strongly to the spike proteins of this particular virus and then dispose of it in a number of different ways. Now, some of these viral proteins are chopped up into short peptides, about 10 amino acids long on average, and they are displayed on the surface of infected cells in complex with human proteins called MHC or HLA proteins. We each have 6 to 12 types of these MHC proteins, but there are thousands of variants in the human population, and different MHCs tend to bind somewhat different peptides. T cells are also important players in adaptive immunity. Again, we have about 100 billion T cells, and most T cells have a receptor on their surface that is distinct from that of others. And if the receptor on this particular T cell can bind sufficiently strongly to this viral peptide, it gets activated, it begins to multiply, and then it can coordinate an immune response in a number of different ways. For example, certain kinds of T cells called killer T cells or CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, It can secrete products that will kill this infected cell and the virus particles that it harbors. Now, after you've cleared an infection, most of the B cells and T cells that multiplied in response to this particular infection die. But a few remain as so-called memory cells, which can mount robust and rapid responses upon reinfection with the same pathogen. This pathogen-specific immunological memory is the basis for vaccination. With a vaccine, you try to induce memory B cells, T cells, and antibodies that are specific for the bug against which you wish to protect the host. Now, I'm going to tell you or recount the history of vaccination by first describing how we conquered a scourge that had plagued humanity since antiquity. And this scourge is smallpox. This was a frightening disease. It began with fever, malaise, and muscle aches. But a few days later, a rash and then pustules developed in the mouth, which rapidly spread all over the body. About 30% of those infected died, and those who recovered were often left with severe scarring. Now, smallpox wreaked havoc across the world. It probably entered the human population about 12,000 years ago, was evident in Egyptian mummies. And just to give you some examples of the havoc it wreaked, between 1868 and 1907, just in one country, India, 4 million people died. In the 20th century, globally, smallpox killed 300 to 500 million people, with about 50 million people infected regularly as recently as 1950s. Now, the Americas were not spared from smallpox. Smallpox was brought to the Americas by the Spanish in about 1520, and it is estimated that over 60% of the indigenous population succumbed to this virus. Boston, where we are right now, had several epidemics. For example, in the 17th century, between 1636 and 1698, there were 6 epidemics. And the 18th century epidemic was a severe one that then spread to the other so-called colonies of the time. The Boston and New York epidemics in 1901 to 1903 led to quarantine facilities. And in Boston in particular, there were 2. One was in Southampton Street, and the other, if any of you have ever gone whale watching off the coast of Boston, you may have seen Gallup's Island, which is where the other quarantine facility was there. And it is also interesting that it is this time that for the first time in the United States, we had vaccine mandates in the Commonwealth of Massachusetts. Now, we don't have smallpox epidemics anymore. Well, why? Well, that all begins by describing an observation that our ancestors made. For example, during the plague of Athens in 430 BC, Thucydides noted that those who recovered from the plague could safely serve as caregivers of the sick. And there are many other such historical references. The Chinese and the Indians then harnessed this observation to protect people from smallpox. And the procedure they developed was called variolation. The Chinese version of this, which was being practiced as early as 1500 AD, went something like this. They collected scabs from individuals who had a mild form of smallpox disease. They converted this to a powder and stored it for about a month. And then this powder was inhaled through a silver tube. And for reasons I don't know, the left nostril for females and right for males. The Europeans encountered a very similar procedure being used in India in the 17th century. There, they collected fluids from the smallpox scabs, stored for a while, and then they mixed it with cooked rice to form a paste. And then punctures were made in your skin, which were then covered with this paste, which is shown here. Now, variolation came to Europe because of a remarkable woman, Marywortley Mountmontague. Her family had suffered a lot from smallpox, and she herself had been afflicted with the disease. She was the wife of the British ambassador to Constantinople, And there she saw this technique of variolation being used. Because she was so afraid of smallpox, she had the British physician there variolate her son. She went back to London, and in 1721, there was a major epidemic, and she had her daughter variolated. Now, meanwhile, the child of the Princess of Wales fell sick at the same time. Turned out not to be smallpox. But nonetheless, she was concerned about smallpox, and Montague, Maitland, and others promoted this process to the princess. And then what ensued was likely the first organized clinical trial, albeit carried out in a fashion that today we would deem correctly both unethical and statistically unsubstantiated. So the trial first began with 6 condemned prisoners in Newgate Prison. And they were told that if they went through variolation, they would be freed. And there was a lot of interest in the press, and these 6 prisoners were variolated in August of 1721. 5 of them developed symptoms and recovered. The 6th one did not develop symptoms, and then it was discovered that this person had smallpox the year before. Now, one of these 5 individuals who had recovered was sent to care for a sick child. And in spite of sharing a bed with this child who had a severe case of smallpox, this person did not get infected. But the princess wanted to variolate her children, and the prisoners were adults. So another clinical trial ensued, and this time 5 orphans were variolated in April of 1722. There was no harm, and then Princess Caroline had 2 of her daughters variolated, thus popularizing this procedure amongst the upper echelons of British society. But variolation was a very dangerous procedure. It often led to serious or lethal illness. Localized smallpox outbreaks would happen as a consequence of variolation. And it is because of this danger, and also the trauma associated with the process where people were bled and left alone in a room after variolation. And for these reasons, variolation was not practiced much. So we did not get protection from smallpox on a large scale because of variolation. All of that changed with the work of Edward Jenner. Jenner was born in May 1749 in Berkeley, in England, not Berkeley, California. And after his training with a surgeon, John Hutton, he went back to Berkeley to start medical practice in 1773. Now, cowpox is a disease that infects cows, but is harmless for humans. And it was often transmitted to milkmaids when they milked infected cows. It is said that a milkmaid told Jenner that she did not respond to this variolation procedure because she had cowpox before. And it is also said that Jenner heard a similar story about a young boy from a medical colleague. So Jenner decided to research cowpox outbreaks. It took a while because cowpox outbreaks were infrequent, but ultimately, after his research was complete, Jenner was ready to do an experiment. And on May 14, 1796, using the variolation procedure, but not with smallpox products, but with pus from cowpox sores, he administered this treatment to a boy called James Phipps. Because this is such a remarkable moment in medical history, we know that the milkmaid's name was Sarah Nelms, who had been infected by a cow called Blossom. Now, 2 months later, Phipps was variolated, but this time with smallpox, and he saw no symptoms. Jenner was elated, and he wanted to publish his findings in a journal called the Philosophical Transactions, which, by the way, is the longest running journal that is still published by the British Royal Society. Now, Jenner was a fellow of the British Royal Society, but the Royal Society refused to publish his paper because they said that he would do his reputation a lot of harm because he didn't have enough data. Nonetheless, Jenner then published his findings through the private firm of Samson Lowe and made some money in the process. Now, Jenner's finding was a paradigm shift because you didn't have to inject people with smallpox anymore. You could find a related pathogen, such as cowpox, which caused no disease when you injected it, and so was safe. But if it was related, its proteins were sufficiently similar to the smallpox virus, which was infecting humans and causing disease. So the antibody and T-cell responses that you got were still protective against smallpox. Jenner, of course, knew nothing about microbes or immunology, but that is why his procedure was safe and efficacious, and it provided a real paradigm shift in vaccine development. So Jenner's vaccine was used widely, and by the early 20th century, smallpox was eradicated in Northern Europe, and only a few cases were reported in the rest of Europe. By 1960, smallpox did not exist in the Americas, and in 1958, the WHO led an international effort using a procedure called ring vaccination aimed to eradicate smallpox from the planet. And indeed, in 1979, smallpox was eradicated. The last known case is a woman called Janet Parker, who was accidentally infected in a lab where she was working as a medical photographer. And in 2017, all vials of smallpox in the world, to our knowledge, were destroyed. And so it really does not exist on the planet anymore. Now, it is important to note that it was possible to eradicate smallpox from the world because smallpox did not circulate in animals, only in humans. It's a much harder challenge when the virus family also circulates in animals, such as coronaviruses, influenza, etc. Jenner is rightly celebrated because his work led to the end of a disease that had plagued humanity since time immemorial. Again, as I said, Jenner knew nothing about the fact that microorganisms cause disease. That had to wait till the 19th century when the modern era dawned. Actually, the foundation for that discovery that microorganisms cause disease was really laid by Van Leeuwenhoek. He was a Dutch businessman and amateur inventor, and he built the most powerful microscopes of his time, which were actually very tiny, about 2 inches long. And he started observing things like the stinger of a bee, mold growing on bread, and such things. But then he also looked at water, both from ponds and so on, and he noticed little species swimming about, and he called them animalcules. Of course, what he was really observing were bacteria. Now, he couldn't observe viruses because viruses are much smaller than bacteria, and his microscopes were not powerful enough. Nonetheless, these bacteria cause severe diseases like syphilis, tetanus, tuberculosis, and so forth. But Leeuwenhoek did not know about that these were bacteria, and of course did not know that they cause disease. That had to wait until the 19th century and the work of two great scientists who were also bitter rivals, Louis Pasteur in France and Robert Koch in Germany. Koch was a precocious child and a brilliant student and became a physician scientist and made very important discoveries. One of the very important things he did was he learned how to grow bacteria in the laboratory, or what is referred to as he learned how to grow bacteria in culture. And then he developed the Koch's postulates for identifying that a particular microorganism causes a disease. The procedure went as follows. The microorganism must be present in every instance of the disease. The microorganism must be isolated from the human, grown in the lab in culture. Then you infect an animal with that microorganism. It must cause the same disease. And then samples from the animal in which the disease thus occurs must contain exactly the same organism as in humans. This, by the way, is still a process for identifying the causative agent of diseases and thus development of treatments. Now, Pasteur, on the other hand, was a very indifferent student as a young man and had quite a path to success. But as a scientist, he made some very important discoveries and inventions. For example, he was the first person to discover chiral molecules, which are molecules that are mirror images of each other, such as certain sugars. He also invented the process of pasteurization, from which we all benefit today. But all of these findings have been overshadowed because of a paradigm-shifting discovery that he made about vaccination. It was a serendipitous discovery indeed. Pasteur was working with chicken cholera, and he injected bacteria into chickens, chicken cholera bacteria, and they did not fall sick. He then discovered that his culture process had gone wrong, and so the bacteria were not functional bacteria. So he cultured the bacteria properly again, and then to save money, I suppose, he re-injected the same chickens in whom he had injected the faulty bacteria. The chickens did not fall sick again. Pasteur has been known to remark that in the field of observation, chance favors the prepared mind. His mind was definitely prepared because he immediately understood that a weakened or attenuated form of the pathogen could confer protection against the fully loaded form of the pathogen, but would also be safe. Interestingly, if you hearken back to the process of variolation, you will see that that is what inadvertently they were trying to do with all this drying and waiting for a month and so forth, but they just didn't know how to do it right in many cases. In any case, Pasteur took this observation and ran with it, and between 1879 and 1880, he advanced this concept and developed vaccines for chicken cholera and anthrax. But he was dying to make a vaccine that would work in humans, and that opportunity came with rabies. Rabies is an infection of the nervous system, the brain, spinal cord, etc., and usually occurs from dog bites or more rarely bat bites. The symptoms include paralysis and fear of water, and so it's also referred to as hydrophobia sometimes. Now, rabies is caused by a virus, so Pasteur could not follow Koch's postulates or even grow it in culture. However, what he did do, he was fearless, and he extracted saliva from a rabid dog. And then, with his colleague Emil Roux, he attenuated the virus by multiple passages through rabbits and drying the spinal cords of these rabbits and infecting rabbits again and so on. So what he was really doing was the following. This virus had adapted to work very well in dogs and humans. Rabbits are not humans or dogs. So by passaging the virus many times through the rabbits, what he was trying to do was to make the virus adapt to the rabbit, which would make it less dangerous for humans. And this drying of spinal cords was aimed to do the same thing. Now, Pasteur showed that dogs could be protected by the virus that he had weakened in this way. He tried it on sick patients. It did not work. But he realized that the onset of symptoms usually lagged the bite. So although he did not know about the immune system, he thought, you know, if I vaccinate immediately before symptoms kick in, then we can hope that the protection mechanism will kick in and protect the person. In 1885, a boy called Joseph Meister, who was bitten by a rabid dog, was vaccinated early, thusly, and it was a success. Pasteur became a hero because of this, developing this process. And it is interesting that Meister was so grateful that he spent the rest, later as an adult, he was the caretaker at the Pasteur Institute that was started in Paris. Now, Pasteur's work really, in the 19th century, identified that one could use these live, attenuated, or weakened pathogens as a vaccine. They were safe, as I said, because they didn't cause full-blown disease, and they were definitely effective because the proteins were the same as that of the fully loaded virus. Later in the 19th century, people also developed inactivated vaccines. These are killed viruses or bacteria, and they are killed so they are safe because they cannot cause disease. And the killing had to be done just right so that you didn't do too much damage to the proteins of the virus, so that the antibodies and T cells that were elicited by this particular dead pathogen were still specific for the real alive pathogens' proteins. In the 20th century, these two types of vaccination procedures developed in the 19th century were in competition and could be compared. And that is the remarkable story of the development of the polio vaccine. Polio, again, is an ancient disease that has evidence in Egyptian mummies. And the central players in the story I'm going to tell you now about the development of the polio vaccine are Jonas Salk and Albert Sabin, both of whom were graduates of the NYU Medical School. What enabled the development of these vaccines is really another major advance in the 20th century, where in 1949, in the 20th century, there were major outbreaks of polio that began to occur with regularity, especially in nations like the US and Scandinavian countries. If someone's interested in the end, I can comment on why these countries with better sanitation were the sites of the big polio outbreaks in the 20th century. They were relatively dangerous. New York City epidemic in 1960 resulted in 27,000 illnesses, 6,000 deaths, and in many cases, that one would see paralysis. So that was the scary part. Suddenly your child would be paralyzed. And in 1921, Roosevelt, who had just been defeated in the election for the vice president of the United States, who was 39 years old at that time, was infected and partly paralyzed. And he started the March of Dimes, an organization that still exists, with the goal of developing a polio vaccine. And as I had started to say, it was really enabled, the development of this vaccine, by an advance that happened in 1949, when Enders, Robbins, and Weller at Harvard learned how to grow poliovirus in the laboratory. Salk realized the potential, quickly scaled up production of this process, and then using formaldehyde, he carefully inactivated the virus, that is, killed the virus, but the proteins, etc., he thought were still intact. With support from the March of Dimes, he carried out what we would call a phase 1, phase 2 clinical trial, which took 3 years, and he determined that the vaccine was both safe and that it elicited antibodies. By now, he knew about the immune system. And so in 1953, Salk announced that he was ready for a large-scale clinical trial. The trial was a tour de force in organization. 2 million children between the ages of 6 and 8 were enrolled. Trials had to be finished by the end of the 1954 school year. And IBM got involved in analyzing the large amounts of data that were created. And on the 10th anniversary of Roosevelt's death, the March of Dimes announced that there was a successful polio vaccine. Salk became a national hero, and people lined up to get the polio vaccine. Sabin was watching all this with mixed emotions. He was the person who had identified that polio infection happens in the intestine and had made many other discoveries about polio. Sabin spent years attenuating the virus, that is, creating a live attenuated virus, not a killed virus. And he did so much like Pasteur by passing it through animals, but also now cell lines in the more modern era. By the time he was ready to try his vaccine in humans, the March of Dimes said that the US already had, and the United States said, we have a vaccine, we don't need another one. It is a remarkable story that at the height of the Cold War, Sabin's vaccine really took hold in the Soviet Union. Millions of people participated in a clinical trial in the Soviet Union. The Soviet Union was the first to begin manufacturing Sabin's vaccine. And then finally, it was also approved for use in the United States in 1961, and interestingly replaced Salk's vaccine in 1962. And the reason it replaced Salk's vaccine was that it was in some ways a superior vaccine. You see, Salk's vaccine had to be injected. Sabins vaccine was taken orally. You just put a few drops of this live attenuated virus solution onto a sugar cube, and then the child ate the sugar cube. That's how I was vaccinated against polio. It also turns out that Sabins vaccine made a kind of antibody called IgG, which protected the blood and therefore prevented paralysis and infection of the nervous system. But Sabins vaccine elicited both IgG antibodies and IgA antibodies. And the IgA antibodies could protect against infection of the intestine because IgA protects the surfaces. A Salk's vaccine still allowed infection of the intestine, and so the feces of a person could still go into the water system and infect others. It is Sabins vaccine and its large-scale global use that has led to the near eradication of polio, except in some endemic areas like in Pakistan, Afghanistan, and a couple of other countries. But then Sabins vaccine is a live vaccine, so a live virus. So over time, this virus in the vaccine could replicate and undergo mutations and become dangerous again. Indeed, polio cases outside the endemic areas are now largely due to such mutations. Therefore, some countries, like the United States, have returned to using Salk's vaccine. It is remarkable, and we should really be so grateful that we have all these effective vaccines against COVID-19, because as time ensues, perhaps, like in this story, we will learn that over different timescales, different vaccines are better or worse. Now, in the 20th century, a new kind of vaccination procedure started to be used. And these are called, these are called subunit vaccines, and really emerged because of the development of recombinant DNA technology. Just as a reminder, recombinant DNA technology is the following. You clone a gene that codes for information for a particular protein. You insert this gene into another DNA of another bacteria or some species or some cell, and then this cell is converted into a factory that produces a lot of these proteins. So people thought, why do we have to vaccinate with real pathogens? We will just vaccinate with the proteins that are important for eliciting a strong immune response. And that's all we need to do. It will definitely be safe because there's no virus or bacteria in it. And it will be easy to manufacture because there's no laborious process of growing viruses, attenuating them just right, and so on and so on. And so these subunit vaccines began to be made, and there are quite a few of them. I'll mention only one that is very successful, and that is the human papillomavirus vaccine, or HPV vaccine. HPV can cause cervical cancer. And what is done is you mass produce the spike protein of this particular virus. And it turns out when you do that, the spike proteins aggregate and form a structure akin to the array of spikes on a virus. And so antibodies can bind with both arms and get a lot of affinity to this. And that leads to a strong antibody response that is highly protective. Well, now we are in the 21st century. And the 21st century, in particular, the desire to combat the COVID-19 pandemic brought us to new kinds of vaccines, viral vectored vaccines and RNA vaccines. Both of these types of vaccines are essentially different from all vaccines that came before, because all vaccines that came before gave either the pathogen or the proteins of the pathogen were administered to the person. In these vaccines, for example, the viral vector vaccines are the ones that are exemplified by the AstraZeneca and Johnson & Johnson the vaccines, and the RNA vaccines are those that are exemplified by BioNTech and Moderna. And both of these classes of vaccines, they do not inject any protein or virus or anything like that. They inject information about the virus. So they are really bringing together, if you like, of information technology and biotechnology. In the RNA vaccines, the information that is encoded is in the form of RNA. Now remember the central dogma, DNA to RNA to protein. So here, what is included in the vaccine is the RNA, the messenger RNA or mRNA that encodes information by the spike of this for the spike protein of SARS-CoV-2. This is encapsulated in the lipid nanoparticle, and these nanoparticles are designed to be preferentially eaten up by our cells. Inside our cells, the RNA is then translated to proteins by our own cells' machinery. And then the proteins elicit an immune response that results in protection from natural infection. It is important to note that this RNA is quickly degraded because cells have many, many ways to degrade RNA, so it doesn't hang around long. The adenovirus vectored vaccines encode information about also the spike protein of SARS-CoV-2 in this particular case, but not as RNA, but as DNA. So what happens here is you take a virus like adenoviruses, which cause diseases like the common cold, for example, and they are engineered so that they cannot replicate. And then the DNA corresponding to the spike protein of SARS-CoV-2 is inserted into the genome of this adenovirus. Now, while the adenovirus is engineered so that it cannot replicate, its spikes, etc., can still bind to human cells and therefore infect human cells. The DNA thus enters our cells, the DNA corresponding to the spike protein of SARS-CoV-2, and then it is converted to the virus's proteins using our cells' machinery to both transcribe the RNA and then translate it into proteins. Thus, we get antibodies and T cells elicited that can protect us from natural infection. Now, again, important to note, the advector cannot replicate and therefore does not cause disease, and infected cells are quickly killed by our immune response, so they don't hang around in our body very long either. So with the success of these vaccines and others to come using similar technologies, the question that we can ask is, are we done with vaccine development? Well, we do not have effective vaccines against highly mutable pathogens. HIV is a prominent example where no vaccine exists. We do not have a vaccine against influenza that can protect against diverse mutant strains. That is why we have to take a shot every year, and it's only partially effective. SARS-CoV-2 mutates much more slowly than influenza, but if global vaccination drives are truly successful, there will be an enormous selection pressure on this virus to evolve mutations that evade the vaccine-induced and natural immune responses that are protective today. And it may be that we get back to the case of influenza or something similar. Well, what we really need to eliminate such scourges from the planet are universal vaccines that are variant-proof, if you like. And many across the world, including my own lab, are working to pursue this quest for universal vaccines. Thank you very much. Those were my prepared remarks, and I'd be happy to answer any questions.
Thank you very much, Arup. That was a wonderful lecture, and yeah, indeed, we have several questions here that, if you have the time, we would like to pose to you. I mean, one question is, how did WHO, the organization, make sure that even small rural populations in remote areas were vaccinated against smallpox? And is there any lesson we can learn for COVID-19 vaccination?
Yes, I can tell you how it worked. And yes, I think there are lessons. The first thing that happened was that was true global cooperation on this issue. that we do have to eliminate smallpox from the world. The vast majority of countries bought into this. And the way they did this in the rural areas and so forth was by deploying large numbers of people, and the vaccination procedure that was used is something called ring vaccination. That you would, what it really means is that you would vaccinate, you would sort of form a ring around the population, and everybody there would be vaccinated. And then you would go to another place. So rather than trying to just distribute it widely, they went location by location and doing it that way. So I think the main lesson to be learned of what can be done if one does this way is really that there was immense global cooperation. And if that happens, it will happen for SARS-CoV-2, but we will not eradicate it from, I mean, I shouldn't say that. I think it will be very difficult to eradicate this virus from the planet because its relatives circulate all over the animal population.
Mm-hmm. And in that there is a relating question, I mean, that is with regards to previous coronaviruses. Do we really historically understand whether human common cause coronavirus like OC43 or others caused pandemia and whether they were attenuated through passing through the population? And is there any expectation for SARS-CoV-2 to pass through the same?
To be honest, I don't know whether these other 4 common human coronaviruses started as pandemic causing diseases and then attenuated. I just don't know that, but I would not like to speculate about that. But I will say that it is interesting that our responses to these viruses are not very long-lived, because that is why we keep getting infected again and again. And so that is a question that I think is well, these days, $1 million is not considered a lot of money. So let me say $10 trillion question, that what is the origin of the duration of immune memory? I think it is one of the most outstanding questions that we need to answer in order to develop the vaccines of the next generation. So I've not quite answered your question because I don't really know whether that was true.
No, but you put a very important question across, that is understand durability of vaccines and how we can do that.
I think it's a critical question, yeah.
Edward has a question that is related to that. And you show very nicely the history of different human vaccines. And he's asking, what do you see in the future of a vaccine technology? Are there new other pipelines coming out soon? Or
So I see vaccine technology to move towards these, in my view, 2 ways. One, I think we will really learn how to develop universal variant-proof vaccines. And the way that will happen is by, in my view, personal view, the way that will happen is by bringing together technologies from diverse approaches, diverse disciplines. For example, we today can sequence viruses, highly mutable pathogens, very, very well. We can then bring to bear sequencing, structural biology, computational analysis to really understand the mutational vulnerabilities of these viruses. And then we can design vaccines that raise immune responses that are not hammers, but are really scalpels that go and attack the virus exactly at the points where it cannot mutate. There will also be, I think, great improvements in the delivery technologies of how we deliver these viruses, these vaccines, in a way that will raise the innate immune response in exactly the right way. And I think there again, our understanding of how vaccines modulate the innate immune response so that we get a proper adaptive immune response is not understood very well. And we are going to, I'm sure, make advances in that. And then finally, I would say that I'll go back to my question that I raised earlier. If we learn how to control the durability of protection and immune responses, I think that will be the next other advance. So those are the advances that, and by the way, they're all happening. right now, people are working on all these questions.
Arup, a question that you left in your talk open and is quite interesting is why countries with better sanitations were sites for the outbreaks? That doesn't sound correct. It sounds counterproductive.
Which one?
Why countries with better sanitations were sites for outbreaks?
For polio,
Yeah.
yes. It's a very interesting thing. Here is what is believed happened. Polio is a disease that is is a waterborne disease. Okay, it is spread by basically fecal contamination of water. Okay, and so it is believed that infants were infected all the time with polio before there was good sanitation because the water was not clean. Okay, and they were immediately protected because the mother had been infected and had before when she was a child, and so during breastfeeding, antibodies for that were passed to the child. So the child was protected by the mother's antibodies. And then what happened was that the child also developed their own de novo immune response upon this infection, and so was protected for life. Now, when better sanitation came along, then most kids or infants did not get infected with polio. And once they were toddlers or older, they were no longer infants. The mother's antibodies were not going to protect, even if the mother had that. And so now when you got it as an older person, you had never seen this disease and you'd had no protection for it, and you suddenly got this disease. It was sort of like getting SARS-CoV-2 now. And so that is what is believed. And I hope I don't offend anything, but that is another reason why in the US and Scandinavian countries, the polio infections were more common in wealthier families, which were probably living in more sanitary conditions.
And given the time, I pose to you a last question, Arup. It is known why the Salk and vaccine, given that they were having essentially the same protein contents, were so different in terms of promoting the response.
So this is something that is, again, not understood, I think, but the difference between them was one very important difference. One was a live attenuated virus, and the other was a killed virus vaccine. And again, I think that, by the way, the Salk vaccine also was not very durable for memory, and the Sabin vaccine was. I did not mention that during my talk. And I think, again, a live virus, even weakened, can raise an innate immune response that is very different from a killed virus, I think. And that may be partly the reason why you got a very different distribution of antibodies and memory and so forth. But these are open questions. So Facundo may know more than I, but these are open questions of why that happens.
Arup, on behalf of all of us, I would like to thank you for a wonderful lecture and a very beautiful story on pandemias. The COVID is not the only one. And yeah, thank you so much for your talk today.
Thanks very much.